TWiM listeners, do you have science you want to share? Microworld.org is the best place to post your microbiology-related news articles, pictures, videos, papers, and more. Sign up for a free account at microworld.org/join and start reaching thousands of microbiology enthusiasts just like yourself.
This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 83, recorded on July 17th, 2014. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michele Swanson.
Hello.
How's it going out there?
It's going great. It's art fair time in Ann Arbor.
Yeah.
So we have more than 1,000 juried artists for the next 4 days, and they bring 500,000 people to our town to see their beautiful work.
500,000?
All at once?
All at once, yeah?
Not all at once. It's spread over 4 days. But
Still
there's music, food, plus all the art. It's a happening.
Do you go?
I do. I was there last night. I strolled for a bit with some friends.
Nice. Well, that's great. And the weather is beautiful there?
Perfect.
Huh.
It's like mid-70s, cool, breezy, sunny.
Also joining us today from Small Things Considered, Elio Schaechter.
Hi there.
How are you?
Very well.
72 and sunny, right?
Yeah, of course. San Diego.
San Diego.
Dry San Diego. But, we're worried about the drought.
It's a big problem, sure.
Huge.
Yep. I don't know what's going to solve it except torrential rains for months.
I don't know. Desalination, maybe. I don't know.
Well,
takes a lot of energy.
There's good things about California, and there are bad things.
Yeah.
You have to take both. Well, also joining us today from the Medical College of South Carolina, Michael Schmidt.
Hello, everyone.
How are you?
I'm doing well.
Here, folks, it is 27 degrees Celsius, but in our building, the air conditioning is broken.
Oh, no.
Just on my floor and the floor below, unfortunately.
Was that Tom Manianis doing?
They're out to get you.
No, nobody wants to get me. Nobody even knows who I am. It's not a problem.
That's just not true.
Yeah, it is. Around here it is, Michele. You would not know. The dean doesn't even know who I am. Anyway,
Keep it that way.
it's been broken for a week, and it is like 95 F in the lab. My students are just dying in there. The incubators are beeping. All the computer fans are running like crazy. And I had to go in today we had a fire department violation in my lab, and we had to remove all the boxes on the top shelves because they were too close to the sprinkler. So I did it because I didn't want my students. Man, is it hot in there.
Oof.
I don't personally mind, but other people mind. I have this Mediterranean genetics that I don't mind heat at all.
So do you have a fan at least?
We don't have any fans, no. In fact, in a BSL-2, you're not supposed to have fans,
Yeah, but in your office with your Skyping.
I don't have one. I actually found an old one in the lab, and it was broken. It didn't work.
Well,
So-
we'll have to say really cool things today.
Say really cool things. Absolutely. Don't get me riled up. I'm really riled up already anyway. There's stuff going on in science that has got me so riled up, but we'll save that for TWiV tomorrow.
Oh, so we miss out. We have to wait till next week.
Oh, I'm telling you. I mean, people are going crazy about regulating science. I just don't understand why. All of us, the CDC has had some issues in the past weeks, of course, but now you have everyone calling, no more select agent work. Let's cut back on the experiments we do. I think it's really scary that
It's short-sighted.
people want to do. Yeah, it is short-sighted and narrow view. Science needs to be, free pretty much, because you never know where the next big thing is coming from. So, you say you shouldn't do this experiment or that. How do you know? How do you know it's not a good experiment? So I get really, I don't know why. I wish I couldn't get so riled up about this stuff, but I do.
Well, you have a nice platform here on the TWiV and TWiM.
Yeah, I do. And I do use it. But, there's some people that should be listening, but they don't, so.
Yeah.
I spoke to a reporter this morning from the UK and he wanted to get the other side. So I hope he will publish that. Anyway, on to cool science. We have 2 papers today for you. And the first one is it will be presented in a lucid manner by Michael.
It's really a very interesting paper. It's entitled Fluorogenic Probes with Substitutions at the 2 and 7 positions of cephalosporin are highly BLA-C specific for rapid Mycobacterium tuberculosis detection. Before you run for the hills thinking that this is going to be all chemistry, it's not. This is really a paper that has something for everyone. And it was authored by Yongfang Chen, Zexen Zi, Pretty Sewell, Hani Hausana, Edward Gravis, Ying Kong, Jeffrey Cirillo, Zhang Hong, and Zhang Hong Rao. And they're affiliated with a number of institutions, starting with the molecular imaging program at Stanford University, the Department of Microbial Pathogenesis and Immunology at Texas A&M Health Science Center, and the Department of Microbiology, Immunology, Biochemistry at the University of Tennessee, and finally at the Houston Medical Center. And this is a really neat paper that caught my fancy. It came out on the ASM Press pick list every week. The press office at the American Society for Microbiology issues the hot papers that came out the previous week. And this one made the list.
You'll find it on their website, right?
You'll find it on their website, which we'll put into the show notes. And
Good.
This paper is published in, and I'm not going to try to pronounce the German name of the journal.
Angewandte Chemie.
Applied Chemistry from the German Chemical Society.
This is what's left of the great achievements of German chemistry in German. The only thing that's left is the title of the journal.
That's
Everything else is in English, including the journal.
So the bottom line of this really neat story is this work first validates the use of an enzyme that's unique to Mycobacterium tuberculosis, which is this beta-lactamase C, which is specifically expressed by MTB, and folks have begun to use it as a biomarker. That is to say, you can use the presence of this enzyme and you feed it a substrate, and then you ask the question, was the substrate cleaved? And if so, you can make a reasonable assumption that MTB is present. Secondly, they developed a specific and sensitive probe, and they named it CDG3 to detect the product of this particular biomarker. And thirdly, and this is where the chemistry comes in, the CDG3, they made some chemical substitutions, and principally what they did is they tinkered with a cyclopropane ring substitution, and this then gave it specificity and selectivity for BLA-C from MTB over all the other beta-lactamases that are out there in the enzyme catalog of the microbial world. And it's very specific in that it's able to detect as few as 10 of their test strains, which was the vaccine strain, which is of MTB, which is BCG, the Bacillus of Calumet and Guerin, which is in the vaccine that's used in a large portion of the globe. And finally, they asked the question that all the clinical microbiologists and public health officials want to know. In a very small pilot study, they asked whether or not this probe that they developed and tinkered with really worked. And the answer
in the real world, you mean?
In the real world. They used real clinical samples. And, of course, everyone was blinded to what was going on. So it was a good clinical trial. So let's start by going, here's how the authors did it, and we'll try to take you through this really neat paper. The authors, if you look at their affiliations, were a mixture of very clever chemists, microbiologists, and clinicians. And what they accomplished in this paper is a really nice story, again, illustrating the interdisciplinary nature that science is requiring in this 21st century. So first up, we have the chemist and the crystallographers collaborating together. Here they exploited the crystal structure of BLA-C. This is the enzyme that's unique to MTB. And they asked the question, what does this crystal structure teach you? And what they learned is BLA-C is bigger, the enzyme is bigger and more flexible in its active site. And the active site, recall, is what takes the substrate. And in this case, it's derivatives of penicillin and cephalosporin are substrates for this enzyme. And they learned that this BLA-C from MTB is bigger and more flexible in its active site. And so this is
than other beta-lactamases.
Than other beta-lactamases. And this was the key insight, or as the patent attorneys will tell you, the inventive step, leading them to the central hypothesis that BLA-C could tolerate a more bulky lactam substrate to provide specificity unique to MTB and thus serve as a unique probe to ask, is MTB present in a clinical sample? So,
can I interrupt you for a second?
Sure.
Michael? It seems to me that this is sort of an illustration of how simple the incredibly tortuous and complex subject of structural biology can be. I mean, this is all you have to, after all the incredible sophistication that goes into figuring out the 3D structure of a protein, you come out with the idea that the active site is bigger.
That's right.
So simple.
This is so cool.
It is.
So the crystallographers and the clever chemists have done their job. They told us it's bigger. So next up are the assay developers or organic chemists. So here they now approach the biomarker by designing an on-off color. We all have seen chromogenic bacteriological media. Most of us use chromager in the clinical laboratories where MRSA has one color, the other staff don't have any color. So we like things with the on-off switch. So they approached this biomarker by designing an on-off fluorogenic probe. So you shine a little UV light on a tube. The ultraviolet light, if the product is present, will fluoresce. And those of us who do real-time PCR know the wonders of cyber green and how real-time PCR is able to give you a quantitation of how much is there. And that's what these authors are exploiting, is the beauty of fluorescence. And they designed their probe to use a single excitation wavelength, and you hit this molecule with the right wavelength of light, and if it's been cleaved, you get this beautiful blue glow. And so you can do this in something as simple as an Eppendorf tube, shine a little UV light on it, and they have a beautiful figure that you can see that shows the glowing. And they started with, and this is where the chemistry comes in, but I thought our listeners would like to learn about the umbelliferone-based fluorescence that they started with. And this umbelliferone is also known as 7-hydroxycoumaron, which is present in many things that we're actually applying to our skin today, namely suntan lotion. And it's
sunscreen.
Yes, that's right.
We use
suntan
sunscreen lotion.
That's right. Thank you, Michele.
From the golfer.
From the golfer. And Michelle's probably used it this past weekend when she was out hitting her
I sure did.
round of par golf. So this molecule absorbs UV light, and it adds. It's a big, bulky, UV absorbing molecule onto the substrate. So again, taking advantage of the crystal structure, this is a big honking molecule. And without getting too deeply into the structural chemistry, which does not lend itself well to a podcast, they show you how they tinkered with it. And they start out with this CDC, what they call derivative, and they move through things. So I'm going to take you through their second figure, where they're actually comparing two molecules, the one that they ultimately select, which is CDG3, and another molecule that has a methyl group attached to it that they call CDG-OMe. And so they have BLASI, the enzyme, present at 10 to 15 moles, and they also have another enzyme from the most common beta-lactamase associated with the gram-negatives, which is TEM1. And in the first panel, what you see is CDG3 and the other molecule, CDG-OMe, and they're cleaved by 10 to 15 millimoles of BLASI, while the one with the OMe side chain is cleaved. It requires 100-fold greater concentration of enzyme in order to facilitate this cleavage. So again, this is illustrating less enzyme, higher specificity. So the compounds, that their chromogenic compound was present in a very low concentration of 10 micromolar. And they also learned that this probe was unable to elicit an activity from penicillinase. So they isolated a penicillinase from common bacillus species, which normally will cleave beta-lactam antibiotics like penicillin and cephalosporin.
And it might be worth adding just that term beta-lactam. It refers to a four-membered ring or square, really, that is the recognized by these beta-lactamases. It cleaves that four-membered ring. So that's where the name comes from.
And which is typical of penicillins and cephalosporins.
Right.
And it's this four-membered ring that if you look at it cross-eyed, it breaks. And, you know, it's all part of the magic that goes with penicillin. So now the microbiologist becomes intrigued that they have a marker or a probe that can detect specific beta-lactamase, and it's pretty specific. If you look at the fluorescence that's coming off of these things, you can see in the panels associated with figure 2, specifically in panel D, how sensitive this BLASI enzyme with their CDG probe actually is. So this is where we now move to the next figure, which is figure 3, where we have real bacteria with the enzyme. So now we're moving from an in vitro chemical sense to an in vivo sense or an in situ sense, where the microbiologist is either taking a culture off of a plate or actually going to the gold standard, which is a clinical sample from a patient. So in panel A of the first figure, they learned that CDG1 is not really all that specific, and it lights up really well. And so when you look at their figures, you can begin to see in the panel A, they have the BLASI specificity. So you have E. coli transformed with BLASI, which is the enzyme of choice, and then you have Enterobacter cloacae, Klebsiella pneumonia with the KPC, Klebsiella pneumonia with another beta-lactamase, and the E. coli with the New Delhi beta-metalloprotease, beta-lactamase in there, and then E. coli with its normal gram-negative. And what you see is CDG1 is not very specific. They're all lighting up. CDG-OMe, which wasn't so hot, is lighting up for just E. coli and a couple of the other gram-negatives with AMP-C or the carbapenemase-resistant Klebsiella. But it's taking about 10 to the 5 bacteria in order to get any fluorescence. But what you see with their probe that they ultimately selected, which is CDG3, the only thing that really lights up with substantial fluorescence is the E. coli with the BLA-C gene in it. And it's lighting up at an extremely low concentration of 10 to the 3 microbes. So 1,000 bacteria are lighting up this particular probe with fluorescence. Then in panel B, they do the standard controlled assay where they put a defined concentration of bacteria in, and they take it through looking at specificity about fluorescence. And again, you don't have to really look hard at this figure because CDG3 is the only thing that is lit up in the presence of BLA-C. All the other strains that have beta-lactamases are dark when you're using CDG3 as your probe. Then they take us through a classic Timecourse asking about timing and enzyme specificity. And what you see is that CDG3 and E. coli with the BLA-C gene, and they have a standard assay of 10,000 bacteria. It lights up fairly rapidly within 180 minutes, and it looks like it's plateauing. But then they also, and this is where the microbiologist gets together with the medicinal chemist to do one of their controls, is they use phenylboronic acid. And phenylboronic acid is a broad inhibitor that binds the serine in this active site of the beta-lactamases that then inhibits the hydrolysis of the beta-lactam. So if you add this inhibitor prior to the probe, you're not going to get any fluorescence. And that's indeed what they showed. And this then enables you to get the sense that it's the active site of this enzyme that is reacting to the probe. So again, this goes back to the specificity of the biomarker. So now the microbiologist is really excited because you got specificity. So now what we all want to know is, will this work with Mycobacterium, which we all know is a pain to grow. TB has generation times measured in days. And, most microbiologists are hypothyroid. We like things to grow very quickly, get our experiments done. And TB requires tremendous patience. So this takes us to the next figure, figure 4. And so now they're going from the bench and they're really invoking a clinical case. So the first panel that they show us in figure 4 is they ask the question, can we take sputum from patients that don't have any tuberculosis, they're TB free, and we ask a question, are there bacteria present in sputum? And we all know the answer to that question is yes. If you can isolate sputum, there will be bacteria present. And then they ask the second question, well, if there are bacteria present in sputum, do they have the ability to cleave beta-lactam antibiotics, penicillin or cephalosporins? And the answer in this particular case is yes. And so we now know that their control sputum does have beta-lactamases. So that's mimicking what we're going to see in the clinic. So then in panel B, what they do is they do a quantitative assay in a classic standard curve where they take defined concentrations of BCG, and they go from 1 bacterium all the way out to 8,000, and they get this tremendous straight line measured against relative fluorescence. And their R value was 0.9824. So this is a standard curve that most folks would kill for. So this is now going to give you the ability to ultimately quantify how many tuberculosis species or how many tuberculosis microbes are actually present in a particular sputum sample. And remember, volume of bacteria is an indicator of disease and the likelihood that the person is going to be infectious or highly infectious, depending upon the number of microbes in the sputum. And finally, in panel C and D, what they show is they show how few microbes it takes to light up this probe. And in panel C, they do, again, a time course. They start with 10 BCG, and they ask the question, do they get fluorescence? And the answer is it's rapid. Within 30 to 60 minutes, you're almost at peak saturation. And then in panel D, they again compare and contrast the specificity of their CDG3 to other mycobacteria and E. coli that have beta-lactamases. They use Mycobacterium smegmatis, and they use 10 to the 7th colony-forming units of this microbe, and they ask, is the relative fluorescence between this non-tuberculosis species different than BCG? And the answer was yes, and the p-value was indeed significant. As you go through their list of looking at E. coli, which has the TEM, which is the classic gram-negative beta-lactamase, Pseudomonas aeruginosa, PaO1, another classic beta-lactamase, and of course, our old friend MRSA, methicillin-resistant Staph aureus, which has a different beta-lactamase activity specific for methicillin. And it, of course, they were all significantly different in terms of their reactivity than the BCG. And again, this illustrates that they designed the right probe that will react specifically to the biomarker that's unique to MTB.
So, Michael, how would you read this, and would you be able to discriminate between TB and these other bacteria in a clinical setting, say?
It's concentration. It's concentration.
So you would need a machine to read the fluorescence, in other words, right? You couldn't just look at it and say plus or minus.
No, you couldn't do it plus or minus, but there are enough instruments out there that measure relative fluorescence, and they measure it in time. And what you're getting is very similar to the types of curves people use often in real-time PCR. And they're looking at the saturation, which is why you see curves like figure 4C illustrating to you how quickly the fluorescence comes up and how it's different than the control sputum that just has the baseline fluorescence, if you will, or the noise associated with it. And
actually,
so,
you call it, I'm a little bit puzzled. You call this fast, but I think that a reaction between a substrate and an enzyme that takes 30 minutes isn't exactly fast. I'm surprised it takes that long.
I think the term fast
does it have to do with the permeability of the bugs or the uptake rate of the substrate?
It's probably getting through the sputum. They're using homogenized sputum, and I don't know that much about assay development in order to optimize it. And the fact that they're bringing in this probe into, these are live BCG. So I don't know the mechanics of how this material gets in, or if the BCG is actually secreting the beta-lactamase. Do you know, Michelle?
I was going to say, I think they're using the term fast relative to the current methods available for diagnosing TB.
That is indeed
not
correct.
In terms of the enzyme, but rather clinical tests. It's really rapid.
Yeah.
And they may be able to, one of the next things you may wish to do is to sonicate the sputum to effectively liberate everything. And that may be the next paper that's in the works where they're optimizing this.
But in practice, you wouldn't want anybody sonicating sputum that might have TB.
Oh, no. That's like a biosafety level 3 no-no.
Right.
And then they'll have people banning that science, and Vincent will be upset again. So now we're at the point with these data, we now have the street credit with the clinical microbiologists, the public health officials, and the people who treat individuals with tuberculosis. You really need to, they're asked to do studies all the time, and, generating a sputum sample is not trivial. You don't just, it's not like swabbing a nose. Getting a sputum sample is hard work. So what they did is they collaborated with, again, a clinical team, and they got 50 blinded sputum samples. And they simply asked the question, could CDG3 detect MTB from this real clinical material that they got from real patients? The sputum samples were mixed thoroughly with transport stabilization solution, and then they homogenated them, and they incubated it at 1 hour before the fluorescence was measured. So it gets to Elio's question of, an hour, which seems to be their optimum. And what did they learn? Well, the tests taught us that it picked up 90% of the true positives, or in other words, the smear positive, culture positive samples, their probe was 10 for 10. And when they looked at the 80% of the smear negatives and the culture positives, or 8 out of 10, were detected by the CDG3.
And
now,
Michael, you might want to just explain that the smear positive means literally you take a sputum sample, you smear it across a slide, and then you look directly in the microscope and ask, are there bugs there or not?
Using a stain that only stains mycobacteria.
Right.
Which is typically the acid-fast stain, which requires substantial finesse. I was going to talk a little bit about that, but I'll do it now. Is the acid-fast stain is very similar in design to the gram stain. It's one of these differential stains. The primary dye, in contrast to the primary dye in the gram reaction, which is
crystal violet.
I'm blanking here for crystal violet. And in the MTB stain, or the acid-fast stain, it's carbofuchsia. So it will develop a fuchsia color. They're affectionately referred to by the clinical microbiologists as red snappers. And then you, of course, have to do a very elaborate heat fixation step where it's resistant to decolorization with acid alcohol. So the TB sputum smears are effectively cooked, and you drive everything in, and it's a technique that requires substantial finesse in order to get it down. And after you do the acid alcohol decolorization step, you then counterstain with methylene blue. And the way I always tell my med students and dental students, it's sort of the backward color reaction to the gram stain, if you want to keep it straight. The gram stains, is crystal violet and saffronin, which is the dark blue purple to pink red. And then, of course, the acid-fast stain is the red/pink to the methylene blue color. And it requires a lot of work. You have to be pretty good at scanning fields, and you need to scan so many fields in order for you to be able to call it smear positive. And there's some debate as to whether or not this old technology is effective at detecting TB, because it requires a substantial amount of work. There are new tests out in the marketplace. The one that has taken Europe by storm is a PCR-based system that uses microfluidics in order to transfer the reagents. And this was developed by a company out of California called Cepheid, and it's called the Gene Expert technique. And it, again, is PCR, and there's a sonicator in the instrument itself, so it does all the processing for you. And it's a really good assay. It's got good performance, but it's very expensive. As you can well imagine, this test will be pretty inexpensive to do, because it's one chemical that you add to sputum, and you can literally shake it. And as the author showed in their figure, if your test tube lights up that beautiful blue after you shine a little UV light on it, and if you think about deploying this technology to the developing world, you can carry a UV lamp in your pocket as a flashlight. We've all been through the TSA lines at airports probably in this country, and every TSA agent, of course, has a UV flashlight that they shine on our driver's license or passports to look for UV-specific inks. And so flashlight technology is much easier to transport in the bush than a Cepheid instrument where you need electricity, you need stable power, and all sorts of other things. So the bottom line to their story is that it was 90% specific and 73% sensitive. And the sensitivity issue, it goes into the analogy that I often use when I'm trying to explain sensitivity and specificity, is specifically you look at belt buckles at airport scanner versus other dangerous metals on planes, of course, box cutters or guns or something along those lines. And this is what we're seeing in this particular case with the Blasi and the Sputum. We have a 90%. It picked up, from the gold standard of the culture positive, smear positive, it was 10 for 10, which is pretty good. It was just in that rare case where they had the smear negative, and if that test requires specific finesse in order to see, and culture positive, the culture positive it effectively missed the smears. And so you look at these things and you say, this really is showing tremendous promise, and that's effectively where the authors leave us as they conclude their paper. They suggest that what we need is more study, and by further validating that this particular probe with a larger set of clinical samples will help them confirm that CDG3 is an important, rapid, and sensitive, low-cost alternative to as a triage test for TB. And again, at the end of the day, why should we care? Well, the facts are tuberculosis has infected a third of our globe. A third of the human population has TB. It's responsible for killing 50% of the individuals that actually have acquired this microbe when it's left.
Wait. I'm sorry. Finish your sentence.
It kills 50% of the individuals when it's left untreated. And tuberculosis in the world causes 2 million deaths, principally in developing nations. And multi-drug resistance is really the biggest demon in the closet. The multi-drug resistance, the extremely drug resistant, and of course the totally drug resistant tuberculosis. And TB is an old disease. It's likely lived with us longer than humans have had language. When you go back and you do the deep sequencing of the MTB genome, they estimate that it's been cohabitating with humans about 70,000 years. And you can actually trace how it has traveled with us as we left Africa. I was always taught that, TB is a disease of domestication. As we domesticated livestock, it came with us, but this predates domestication of livestock. I mean, TB probably came with us as we emerged out of the savannah. And so I think this really would have great utility here in South Carolina, because our jail in Charleston has had an outbreak of TB. Our school system in South Carolina had an outbreak of TB. And, you'd really like to be able to rapidly screen people if you could, get a sputum sample or get a sample that you could then subject to this very simple and straightforward test. So that's why I picked this paper this week. I thought it was a really good example of the collaborative nature of science and how cool stuff can actually happen.
I'm not sure about the specificity here. So I asked you earlier whether you could do a plus or minus test. And you seem to now imply that you could just shine a UV light, and if it glows, then it's TB positive. But some of the experiments here, the fluorescence intensity, there's a difference between TB and other beta-lactamase containing bacteria, but I don't know how that would translate in a test. And the limited number of specimens they have here don't address that. It seems to me that they would need to spike clinical specimens with other bacteria and see at which point you get a false positive or something like that.
I think that's why they use the control sputum. You see the background fluorescence in Figure 4C, the background fluorescence, and if you do a subtraction of the background fluorescence, you can ask the question: is it above baseline? This assay is clearly not ready for the clinic yet. They have to do some further development, but. Yeah. It's at the stage where it's promising, and I think, as you say, what you want to be able to see is a larger number of clinical samples, and the fact that in their in situ tests with 10 colony-forming units of BCG, when looking at the relative fluorescence intensity, you'd have to almost have a chart to see how bright it was.
Yeah.
In order to see what was actually going on.
Yeah, nonetheless, very interesting that there's collaborative effort going on to address an important problem. It's something we haven't really talked about, assay development before, so that's good. Thank you.
And assay development that can work in resource-poor countries.
Yeah.
And clinics, yeah.
Here in the U.S., it's not a problem, right? You can make an expensive assay.
Yeah, relatively.
Well, the Cepheid assay was first pioneered over in Europe, and it's only recently come to the United States. And of course, it's expensive. The instrument itself is in excess of $100,000, and you have to purchase it. And it doesn't require a skilled technician in order to run it. You just effectively load the sputum in. But at the same time, the instrument requires annual calibration, and the cartridges used are not a trivial expense. It's between $50 and $100 a cartridge, depending upon how many you buy.
You mentioned, Michael, 50% fatality rate. We should clarify that that's of people that have active disease.
Yes.
Quite a lot of, a third of the world's population is carrying TB, but people with a healthy immune system are able to confine the microbe to these walled off areas in their lung, and they never become ill.
And I was going to get to that when Elio interrupted me, and then I forgot. And I was going to talk a little bit about the granuloma and the way TB is often taught is that you become infected with this microbe. Your immune system mounts an aggressive challenge to it, and it effectively makes a brick wall in your lung, and it walls it off with calcium, which you can see on an X-ray. If you've walled it off, you can see that you had active disease. But for many people that are healthy and have good nutrition, the immune system basically crushes it like a bug.
So I have a personal story that's relevant. My dad grew up in Italy in the 1920s and was a physician and apparently acquired TB. But lived with it most of his life. And then here in the U.S., when he developed cancer when he was 60, as soon as he received chemotherapy,
ouch,
the TB
came back,
and it ruined his kidneys. So he had renal tuberculosis, and one of them was completely blocked. Yeah, so it was
That's
there.
Pott's disease. That's Pott's disease.
Pott's disease, yeah.
Yeah.
So... yeah, it's there. It was there, and they didn't know it until it reared its head, right?
So there's not routine screening. What would you use, a BCG test or something to see?
No, you can. If you had TB, you're going to be smear positive as long as you have an active immune system, because the TB skin test that we're all familiar with, those of us who work in healthcare get tested each year. And many of our kids are tested when they go to school to make sure they're tuberculosis free.
But if you're about to put somebody on immunosuppressive therapy,
should you
seems
test?
to me you'd test first and be ready with the anti-TB drugs.
It goes for viruses too. Everybody has herpes and CMV, and if you get an organ transplant, they're going to come up, but it's not tested because the assumption is that everyone has it, and they wait until it comes, and then you deal with it. So in his case, they gave him isoniazid, right? But that was the least of his problems at that time.
Tough.
Yep. All right. Thank you, Michael.
You're welcome.
I do think that since this was a paper from a German journal, hats off to the Germans and their World Cup victory.
Oh, that's right.
That's right.
All right. You're going to get email, but that's fine.
Yep.
From Brazil.
They're a great side, great team.
All right. Our second paper is in MBio. It's called Invasion of the, I hate to say body snatchers, but Invasion of the Central Nervous System by Cryptococcus neoformans, requires a secreted fungal metalloprotease. And the authors are Vu, Tham, Uhrig, Chomson, Pombeira, Jamklang, Bautos, and Jelly. And they are from University of California in Davis, San Diego State University, and Davis again. So many listeners want more papers on fungi, so this one caught my eye, and it's a really neat little story, very straightforward, I think, and you'll like it. Cryptococcus is a fungus. It's a common fungus found in the soil that infects people, and it causes a disease called cryptococcosis. And it and another one, Cryptococcus neoformans, there's another one, Cryptococcus gaddii, are the two big ones. There are 30 different species of Cryptococcus, but those are the two that infect people. So it can be found in the soil throughout the world, and you inhale spores basically from the soil, or sometimes from pigeons. They contain it, and you can inhale their droppings, and that will do it as well. And they can cause a lung infection. If you're immunocompetent, usually you're okay, but not always. It can cause disease in immunocompetent people. But people who are immunosuppressed for whatever reason, the fungus can spread from the lung and get into the central nervous system. And that's a big problem, and that has a high mortality rate. So people who are infected with HIV, for example, are at high risk, especially in sub-Saharan Africa, to get serious cryptococcal meningitis. So there are about 1 million cases a year globally, with 600,000 deaths of infection with crypto neoformans. And this paper addresses a question that I've always been interested in for viruses, how the fungus gets into the central nervous system. It's the same thing for some viruses which are able to do that. The CNS is pretty well protected, but some viruses and other microbes can get in. So there are 3 general ideas about how that happens. One is, as everyone knows, there are very, very tiny blood vessels in the CNS, the brain and spinal cord. They're microvessels or capillaries. They're made up of endothelial cells, which have very tight junctions, and they form the vessel. And sometimes if there is inflammation in the region caused by an infection and an immune response, this will loosen the tight junctions, and viruses, or in this case, the Cryptococcus, can get in. A second idea is that the microbe can get into a macrophage, which have the ability to cross capillaries and get from the from the capillary into the CNS. And then finally, a process that I call transcytosis, where the microbe will be in the lumen of the blood vessel. It binds to the endothelial cell. It's taken up by endocytic process into a vesicle. The vesicle traverses the endothelial cell, goes to the other side where it's released. So endocytosis and exocytosis. And that's been proposed for viruses, and there's some evidence for bacterial pathogens that do that, as well as Candida albicans. So maybe Cryptococcus does this as well. And so previously it's been found that cell surface and secreted proteins of C. neoformans have a role in invasion. There are extracellular proteins like phospholipases, ureases, lactases that seem to correlate with brain infection. And there's also evidence that the fungus has to bind to a receptor on the surface of the endothelial cell in order to get into the brain. So that's where this story begins. In previous work, what they did is study the proteome, all the proteins that are on the cell wall or secreted by C. neoformans. So they purify these and they identify each one of them and they ask what's there. And one of the proteins they previously identified is known as a metalloprotease. This is an enzyme that cleaves proteins, or it could be other substrates as well, that needs a metal for its activity. And the metal can be zinc or can be cobalt as well. And another fungus, Aspergillus, has a metalloprotease that's related to the one they identified in cryptococcosis. And that one can break down elastin, which is a component of blood vessels. So you get the idea that this metalloprotease could be involved in crossing the endothelium. So what they do in this paper is ask, is there any evidence that this metalloprotease can play a role in getting into the CNS? And for a good part of the paper, they use a model, an in vitro model of the blood-brain barrier. They take human cerebral microvascular endothelial cells. So these are cells taken from the tiny blood vessels in the brain. They purify them and they're put in culture on a membrane, which is then floating in a cell culture dish. So there's a top and the bottom side. And you have medium on the top and the bottom. They're separated. So the top side represents the blood and the bottom would represent the brain. So what they do is they add cryptococcus to the top, and these cryptococci pass through the endothelial cells and they come out the bottom, and they can take the medium on the bottom and simply count the colony-forming units. So it's an assay for transcytosis. It's pretty cool. So what they do is they knocked out the gene encoding this metalloprotease. It's called MPR1. And they ask, can the cryptococcus cross this endothelial layer? And the answer is, when you take the gene out, it's not very good at crossing the endothelial cell cultures. All right, so you can simply add them and measure the transcytosis. And if you take away MPR1, they're defective at doing that.
You want to say something about how they ran into this gene?
Yeah, I mentioned that they did a proteome analysis previously on secreted and cell surface proteins, and this was one of the proteins that they had identified. And there had been evidence that a related protein in Aspergillus breaks down elastin. Also, in a rabbit model of cryptococcosis, this gene is upregulated during infection of the brain. So there was some evidence that it might be involved, and that's why they did these studies. So next they say, well, for transcytosis to occur, typically the microbe has to attach to the endothelium. So they measured attachment. So they say, does MPR1 play a role in attachment? And the answer is yes. The cryptococci do not attach when MPR1 is deleted. And finally, they ask, is this gene needed for survival inside of, say, a macrophage? Because maybe it's getting in via macrophages in animals. And so they ask, does the cryptococcus survive any differently inside of a macrophage with and without MPR1? And there's no difference. So you don't need this gene for intracellular survival. It looks like you need it to attach to endothelial cells, leading to transcytosis. So now there's some evidence that MPR1 is important for getting into the CNS by crossing the endothelium. They do a cool experiment where they take the gene and they put it in Saccharomyces cerevisiae. All right? In this, Saccharomyces doesn't have a metalloprotease. They put this gene in, they confirm that the protein is produced, and when they add this yeast to their endothelial cell assay, it crosses the endothelial cells.
Amazing.
I mean, SC...
What a great experiment.
On its own doesn't cross.
Yep.
Just put one gene in, and that's amazing. Yeah, it's just amazing that this is the kind of experiment you want, right?
Mm-hmm.
It's the only kind of experiment you want.
It's a binary experiment. You put the gene in.
I had a chance to chat with Kiam Vu, who's the first author, who is a student and now a postdoc in this group, and he came up with this idea, and it was he was motivated by wanting to test whether it was sufficient to mediate translocation. So it's easy to knock something out and say it's required, but then is it sufficient? Is a really tough one in biology. So he came up with this really cool assay of using a non-pathogenic yeast.
It's a good idea.
Yeah, the same idea has been used with bacteria, like taking beta hemolytic strep genes and putting them in a lactococcus, which is about as harmless as can be, and making it into a pathogen, actually.
We call it a gain of function test.
Right.
Oh my, gain of function. It's what people are in.
Loss of function, because you need both.
People are up in arms about.
That falls under the dual use clause.
But in this case, it's fine because Saccharomyces is known to be killed in macrophages. So this was a safe experiment.
Yeah. They did a few more cool experiments. They used scanning electron microscopy to see what physical changes happen when we're back to cryptococcus now, when it attaches to endothelial cells. And if you just look at the yeast cells, the fungal cells themselves, the ones lacking NPR1, they look different. They have a more porous surface compared to the wild-type cells. And then when they add them to endothelial cells, you take the wild-type fungus and you add it to endothelial cells, you get a lot of microvilli produced on the surface. And it's been previously suggested that these have some role in transcytosis. And the mutant cells induce fewer microvilli-like structures, which may reflect or may be a reflection of them not binding as well. Okay, then the in vivo experiments. They have a mouse model for cryptococcal CNS infection. So they put the fungus intranasally into mice. It replicates in the lung and then can spread elsewhere. So they use the wild type and the mutant strains. The mice live a lot longer when the fungus lacks NPR1. At day 37, there are no deaths in mice that have been infected with the mutant fungus versus 87% in wild-type mice. In both, the number of cryptococci in the lung is similar. So you're not affecting proliferation of the fungus, but rather something specifically needed for survival.
They don't make a point of it, but the controls still die.
They do. They do. And, they do die. Eventually they do, yeah.
So, they take longer, but why do they die? I mean,
well, that's a good question, yeah. I don't know.
How does it get in?
I don't know if...
it sounds like there's another mechanism.
Could be.
There.
It's not clear that they're dying from crossing the blood-brain barrier, though.
Yeah.
I think those mutants still have a really high burden of fungi in their bloodstream and other organs. So...
You're right.
I would say that we don't know if it's CNS.
But it seems to me that those graphs are a little bit incomplete, let's say, with regard to interpretation. Or at least there's something interesting going on there that they don't address.
Now, in the brain, so this is relevant to this point, if you look at the fungal burden in the brain, again, the burden is much reduced in mice inoculated with the mutant. So it's not absent. Right, so it's much reduced. So there may be other ways. It could get in by macrophages, right? That would make perfect sense. I don't see that there has to be just one route of getting in.
True.
But in other organs, heart, spleen, and kidney, the burden is similar, mutant versus wild type. Now, if you put the fungus in the tail vein, so now you're bypassing the initial replication in the lung, you put the fungus in the tail vein, wild type, all the mice are dead on day 21, and again, none are dead with the mutant. And again, there's much less fungi in the brain of mutant inoculated mice versus wild type. When they put the fungus directly in the brain by intracerebral inoculation, both the wild type and the NPR1 mutant grew equally well. So, it's taking away NPR1 is reducing its ability to get into the brain, but if you put it in there, they can replicate. They both replicate well. And in the brain of animals that are infected with the wild-type fungus, if you look histologically, you can see lots of lesions full of, in the parenchyma, the brain proper, full of cryptococci, and they're often near the vessels, so they come in through the vessels and begin to proliferate near there. And they see none of these in the animals inoculated with the mutant fungi. No lesions whatsoever. So I think pretty good evidence that this protease is important for getting into the CNS. I had a few thoughts at the end. I really would like to know what the ligand is for the protease.
Yeah.
What is it binding to on the endothelium? And, this metalloprotease is a enzyme. Do you need the enzyme activity for invasion? You could easily knock that out by changing the active site, right? One amino acid change, and ask whether you need it for entry. And then, of course, the real question is, does this matter in people? And I don't know how you would do it other than having an inhibitor of this enzyme and then trying to treat people with it. But you'd have to go through a lot of animal studies before you could do that. Anyway, I thought it was a cool story. I learned a lot. And this is another important disease, especially in developing areas of the world, that requires some active work to take care of it. Yeah.
Yeah, and in general, we don't have great antifungal therapy because yeasts are much more like humans than bacteria are, and so our drugs are not. We don't have fungal-specific drugs, as many fungal-specific drugs. The ones that we do have are pretty toxic to the human as well. So, this is an important area of research. I did have the pleasure of talking with TWiM Vu, the first author. He is, as I said, a PhD student in the lab and then is now a postdoc for a bit longer. He moved with his family to the United States from Vietnam in 1995 as refugees. He then got a bachelor's degree in chemistry at the University of Pacific Stockton, but minored in biology. And he said it was the biology classes that really captured his imagination. And he realized he could ask questions about much more complicated, interrelated pathways if he also pursued biology. So he went to UC Davis and got his PhD there and was quite productive. He was first author on 5 papers and contributed to 2 others, including one to the ASM Journal Infection and Immunity, a Journal of Biological Chemistry paper, and a couple in Eukaryotic Cell. So clearly a talented and ambitious scientist. His expertise was in the in vitro tissue culture models for studying this translocation. And then he had collaborators when it was time to move into the mouse models and to do the histology, et cetera. He also said that what they're excited about is not only is this really important mechanistic work about a fungal pathogen, but they're really intrigued by the possibility that this protease could be used in drug delivery. Yeah. So they're now collaborating with chemists at UC Davis who are synthesizing nanoparticles that they then conjugate on their surface to this MPR1 enzyme. And they're going to test in their experimental system whether they can get those nanoparticles now to cross into the blood-brain barrier, If so, they could then consider using this as a mechanism to get drugs into the brain, which is right now really difficult to do.
Are they going to use a proteolytically inactive version, or?
I didn't ask that, but I agree with you that that's an interesting question. And of course, if they can just identify maybe the peptide that mediates
that
this,
would be best.
Then the synthesis gets a lot easier.
And then there are fewer side effects that you'd have to worry about also,
right,
I guess.
Right. The other biology we didn't talk about is listeners may wonder, why would this fungal pathogen have a molecule that gets it into the brain?
Right.
Yes. Yeah, I think it's worth pointing out that it's not a human. Humans are not its natural reservoir. It actually is very successful in the environment, and probably this extracellular protease can allow the fungus to break down proteins and use that to generate nutrients. So it's an accident, most
absolutely.
likely.
Yes, that's really important, and something we've brought up before about what makes microbes virulent. And here, it's often an accident because of something else that it has to do. So, in the lab, this thing obviously grows well without the protease, but I bet in the soil, you need it for something.
So, Vincent, are there other examples from the virology research community of a single molecule that can mediate translocation across the blood-brain barrier?
Sometimes receptors will do it, as in this. So virus attachment proteins that will bind to endothelial receptors have been identified, And for polio, that's important. It has the receptors on the endothelial cells, and that probably contributes to transcytosis. But
across the blood-brain barrier specifically?
What about rabies?
Yeah, rabies gets in via neural pathways. So, it gets in at the neuromuscular junction via a virus receptor interaction, gets in the cell, and then is transported all the way up into the CNS.
Yeah. But to circle back to the point you made at the beginning, Vincent, about how we need to be open-minded about what research is important, here they set out to study translocation of this fungal pathogen into the brain,
right.
And they may have discovered now they've got a patent on this molecule, and it may open up really new techniques for drug delivery. So
I think this is really important. You never know what's going to come. So to limit what you're studying in any way, it can be a big loss for what we do know. And just because something is dangerous, I don't think we should say don't work on it.
And it's a waste of all that potential.
That's my personal view. I know a lot of people don't agree with that. I think we have to take risks. We have to do our best to mitigate the risk. And I know we're all human and we make mistakes, but I think in research, the benefits always outweigh the risks. But I seem to be in a minority.
I agree with you. I'll
Really?
jump on that. Yeah.
Because a lot of the really, seminal observations have been, sort of experiments that have happened by happenstance or
absolutely.
Accident. And
sure. I mean, the CRISPRs, restriction enzymes, cloning, all this stuff comes from people just following their curiosity and letting the smart people with good labs do it. And right now there's a big move to stop so-called dangerous, quote-unquote, research. Perceived dangerous, but which I think is wrong to begin with. But I don't think that makes any sense. I think that the benefits are sometimes not predictable, maybe often, and you have to do it. So.
And there are ways to reduce that risk.
Of course. And I think reducing the experiments not the way.
And we have 40 plus years since, the IBCs were constituted and, in the 70s out of the outgrowth of the Asilomar conferences. And then the NIH, stood up. The scientists would self-regulate themselves at institutions. And we've done a fairly good job of self-policing ourselves. I mean, we haven't opened Pandora's box and, let anything out.
Michael, you might want to just say what an IBC is.
Oh, an institutional biosafety committee that any institution or company that accepts federal funding is required to assess the risk associated with the science that is going to be conducted in their laboratories and then develop a mitigation plan to control that risk, not eliminate the risk, but to control the risk so that the science is conducted safely and that there are provisions so that any unintended consequence, should it happen, not adversely affect the general public or the institution that the work is being conducted in, causing harm to the laboratorians or any of the other folks associated with that institution.
Okay, let's do a couple emails here. We have one from Matt who writes, "Hello, TWiM team. It was great seeing some of you at ASM last week in Boston." So you can see this is a bit old. Either in passing between sessions or at the live TWiV episode. I have a few questions about the ASM meeting. What was something that you all took away from ASM that was particularly interesting, inspiring, or changed your views about a topic? Well, in fact, we did an episode on that, right? I think
we did.
The week right after the meeting.
Let's refer him to that episode.
So let's, I don't know what it is offhand.
I think our letters, his letter crossed as we put up the episode.
We're behind on our letters.
Yes.
Second, from episode 78, A Bacterium Grows in Brooklyn. There was discussion at some point about the hypoxic environment of the hemolymph impacting the growth of the fungus. That raises a question. I thought the hemolymph was the blood equivalent for insects, and I presumed it would be oxygen-rich. That made me think about oxygen utilization by bacteria in an animal's bloodstream. Blood in animals is oxygen-rich, but is mostly bound by hemoglobin. Are bacteria that end up going septic able to utilize that oxygen bound by hemoglobin, or must they be able to survive in a hypoxic state? I can't remember talking about this in my bacteriology classes. My notes and a quick search around the web didn't turn up a clear answer. It's always a pleasure to listen and learn from all of the TWiX podcasts. Matt is at Washington State, and he's in the veterinary medicine class of 2016. So what do we think here? Can bacteria utilize hemoglobin-bound oxygen? Now, Michael, you must know this, no?
I honestly don't.
Wow.
I was just racking my brain of trying to remember if things like E. coli can steal oxygen or salmonella can steal oxygen from hemoglobin, and I don't honestly know.
Well, we certainly know E. coli can grow in blood.
Yeah, we know it grows in blood.
Yep.
And we know salmonella grows well in blood, and and salmonella is able to steal the iron. But that oxygen is bound pretty tightly to the heme. So it really depends on the how oxygen interacts with the cell membrane, and I don't know the answer to that question.
So they must grow anaerobically then, right?
Yeah, the bacteria grow anaerobically.
Sure. Okay.
Well,
do you know Elio?
No, I don't. But I'm puzzled.
It's a good question. It's a good question.
Do you have a lot of sepsis from Pseudomonas, which is pretty aerobic? I think it does cause sepsis.
Yeah,
it does cause sepsis.
So
But Pseudomonas aeruginosa is thought now to be a facultative.
Is it? Oh, okay.
Everybody is beginning to, the majority is swinging towards facultative. Med students were taught dogma that it was a strict aerobe. And now we're learning more about that wonder bug that is Pseudomonas.
All right.
All right. Well, if any listeners know the answer, let us know. Next one is from John, who writes, Dear recently evolved hosts, that we are. It is 28 degrees centigrade in Grand Junction, Colorado. At episode 75, time 1 hour, 18 minutes, and 10 seconds, Vincent suggests that icky stuff that came out of us probably can't hurt us because it was already in us. Coincidentally, one of my oldest memories is watching a baby trying to eat the contents of its diaper and asking my father what happened if you did that. I suggest listening to a podcast called This Week in Parasitism, where the hosts describe parasite life cycles that depend on recycling waste. Absolutely. We've certainly talked about that. I have two comments on episode 74 between 8 and 10 minutes. One, I was confused by the discussion of which species have endosymbionts. I don't think you were using a consistent definition. Both cows and humans depend on symbiotic gut bacteria. These bacteria like their hosts, but remain potentially free-living forms. They are not obligate symbionts. Is facultative symbiont a proper term? In contrast, mitochondria are obligate endosymbionts.
Well, there are obligate endosymbionts in insects, plenty.
Sure.
Buchnera and Wolbachia Buchnera in aphids, in sap feeding insects, is essential for growth of the aphid. You treat the aphid with antibiotics and they starve to death. So, and they are endosymbionts.
The bacteria in our gut are not endosymbionts, right?
Sorry?
The bacteria in our gut are not endosymbionts.
No, the bugs in the rumen of a cow are not endosymbionts.
So those
symbionts, but not endo.
So are they obligate symbionts? Can they grow elsewhere besides our gut and the cow gut?
Yeah, sure. You have to have very special techniques and be very good at it. But an awful lot of I don't know if all, probably not all, but many of the bacteria in the rumen can be cultivated if you're extraordinarily careful.
Yeah.
Hungate developed techniques where you have to really do everything so strictly and aerobically that an atom of oxygen will screw it up. So,
yeah. Number 2, youth as a species is not an excuse for not having symbionts or other characteristics. Each of us, human, insect, or microbe, belongs to a billion-year-old lineage, but not a billion-year-old species. Individual insect species are of comparable age to humans as a species. We could and almost certainly did inherit gut bacteria from our ancestors the same way extant termite species inherited their bacteria from extinct ancestors. I mentioned termites because coevolution of termites and their symbiotic bacteria has been studied and parallel phylogenies reconstructed. I am far away from my book collection, but I recall this was explained in Evolution of the Insects by David Grimaldi and Michael Engel, which is worth a read in any case, and he provides a link for that book. And
interesting points.
Very good points, absolutely. Jim writes, greetings, micro monarchs.
Oh, we're kings now.
We get new names all the time. Here in Vancouver, it is a wonderful spring afternoon, partially cloudy, 16 Celsius. I know Dr. R is a bit sensitive about sensationalizing science to scare the public, but I must respond with a listener pick based on the horror scenario of TWiM 78's fatal insect fungus sprouting out of human heads. My pick is the audio play Fungus Among Us by Steve Newby and narrated by the supremely sinister-sounding actor Malcolm McDowell. It's available at audible.com. This gem is right on target, mixing a mad scientist with bad science for some good, clean fungus.
Oh.
There you go. Still waiting for the first episode of This Week in Fungi. Well, you had a little excerpt of fungi today, Jim.
And actually, the second paper used, right.
Yep. And one more from John. Dear microbial incubators. Today we are getting very creative with the names, aren't we? It is 40 degrees Celsius in Death Valley. And I confess to writing from here in an attempt to win the weather report.
You win.
win.
I am listening to TWiM during a long road trip. Understanding takes some effort, but I haven't crashed yet. Regarding recent discussion of supplementing antibiotics with bacteria, is it possible to engineer strains of E. coli and other gut bacteria that are resistant to standard antibiotics? Then one's microflora could survive antibiotics. The resistance would have to be a kind that is rare in the wild, but that hospitals are accustomed to dealing with. This would reduce the consequences of trading resistance genes with pathogens. Yeah, I guess you could do it, but then if you ever got an infection from those, you couldn't be treated, right?
Correct. And remember, it's your intestinal barriers that break down, and half of the infections that folks get in the hospital actually come from yourself. It's
they get into the wrong place.
They get into the wrong place. Your own barriers break down. So that would not be good.
So you might have to engineer some sensitivity into them as well. So you can hit them with the second antibiotic. It would be
there's always the suicide gene approach where the antidote is to be present. And, I did about 5 years of work on using suicide-based genes with antidotes in order to control bugs. And when the suicide lost its antidote, the microbe would be depleted from the community. So that technology has been tried, but natural selection always seems to win out.
Hard to get approval.
Yeah. Possible. John, enjoy your drive through Death Valley. And be
Hopefully
careful.
He's through by now.
Yeah, probably, but be careful, right? And this is TWiM 83. You can find it at iTunes and also at microbeworld.org/TWiM. We do like getting your questions and comments, and we love the names that you call us, so keep it up. You can send them to twim@twiv.tv. And I want to thank everyone for participating today. Michele Swanson at the University of Michigan. Thank you, Michele.
Yeah, till next time.
You bet. And Elio Schaechter is at Small Things Considered. Thank you, Elio.
Oh, my pleasure.
And Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you.
I'm Vincent Racaniello, and you can find me at virology.ws. I'd like to thank the American Society for Microbiology for supporting TWiM, and in particular Chris Condian and Ray Ortega for their technical help. The music that you hear at the beginning and end of TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.